Hello, is anyone there?
Yes, I'm here.
Okay, thank you. I want to make sure somebody could hear me and it was working. Actually, earlier today it was not working. Let me share my screen. There we go. So today is August 15th and we're supposed to do lab 9. Any questions about that? Do note you should be working on your unknown project. I'll try and send out results after the lab tonight. You should also note your infectious disease paper is due this Saturday. Any questions about any of that? All right, so let's go to lab 9. I don't think I have it. My computer's a little slow because it's still working on the lesson for the lecture. Remember, when you're turning in an assignment, only turn in the worksheet, don't turn in the lab module. All right, lab 9, identification of gram-negative bacilli. We have lab 9 to finish. You need to finish you need to finish the case studies and then complete lab 9 worksheet. Any question about any of that? All right. So there's some reading, which is normal for the lab, but there's not very much today. Just a few pages on biochemical tests. Actually, only one page there, and then a few pages on classifying and identifying microorganisms. There will be some video clips in today's lab. Upon successful completion of this lab, you should be able to one, interpret the results for the following biochemical tests for the IMVIC reactions. Let me warn you, IMVIC is an acronym for 4 different tests. IMVIC, I for indole, M for methyl red test, V for Voges Proskura test, and then the C for the citrate test. We'll talk about all of those tests today. Also know how to interpret the results for the carbohydrate fermentation test, the motility test, and the hydrogen sulfide production test. 2, be able to explain how to interpret triple sugar iron or TSI test, the SEMS deep test, and the growth on McConkey's and Hectone enteric or HE agar plate. McConkey's is also abbreviated MAC. All of these tests are running more than one test at a time. I think all of these are actually running 3 tests at one time. Maybe MAC is only doing 2, but SEM is definitely doing 3. Triple sugar iron is definitely doing 3 or more, and HE is definitely doing 3. And then 3, be able to use biochemical analysis to solve case studies involving gram-negative bacterial infections. Any question about any of that?
Yes. So if we have a gram-negative bacteria for our unknowns project, would you want us to request, say, the methyl red test individually or as part of the group of tests?
When you're requesting a test, request a specific test. IMVIC is an acronym for a group of tests which typically are used on Enterobacteriaceae. They're not used on all gram negatives. Okay,
Yes, thank you.
all right. So members of the family Enterobacteriaceae are gram-negative bacilli, and they're all small gram-negatives. They're non-spore-forming bacilli, and some of them are so small that they're actually called coccobacilli. They are facultative anaerobes, meaning they do grow best in the presence of oxygen because they can perform aerobic respiration, but they do not need it. They can perform another form, either fermentation or anaerobic respiration. The Enterobacteriaceae is a large family. It has 53 genera in it. 24 of them are known to cause human infections. They are all glucose fermenting, oxidase negative organisms. And here we see some of the genuses in this family. Most of these genera are normal microbiota of the intestinal tract, aka coliforms, although Salmonella is not normally within the human intestines, but it is in the intestines normally of birds. They are opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, and septicemia. Common genera responsible for causing opportunistic infections include Escherichia, including E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several of these genera cause gastroenteritis, and these are frank human pathogens. These include genera Salmonella, Shigella, and then E. coli serovar O157H7. This E. coli with this antigen, actually these two antigens, is a human pathogen, and it's bad news. Rarely does this happen in the United States, but occasionally you'll read a newspaper article that several people were going to, I don't remember, some fast food place, and then they were getting exposed to O157H7, and then they get gastroenteritis. Any question about any of that? All right. The Pseudomonaceae is a family similar to the family Enterobacteriaceae in that they are all gram-negative bacilli. Pseudomonas aeruginosa is the most famous member of the Pseudomonas because it's a human pathogen. The Pseudomonas are all non-glucose fermenting bacteria, and they're all oxidase positive. And so those are two ways you can distinguish the Pseudomonas from the Enterobacteriaceae. The glucose fermentation, not present in the Pseudomonas, and they are present in the Enterobacteriaceae. And then the Pseudomonaceae are oxidase positive, the Enterobacteriaceae are all oxidase negative. The Pseudomonas can be opportunistic pathogens, particularly Pseudomonas aeruginosa can cause infections in immunosuppressed individual, burn patients, and trauma patients. Pseudomonas aeruginosa is very lousy at causing an infection in a healthy individual. And that's simply because it is not good at breaking the first line of defense. You need to have a patient with either the skin injury or a mucus injury, such as a burn patient or a trauma patient. Pseudomonas aeruginosa is also implicated in healthcare acquired infections, and we talked about that earlier as a nosocomial infection. It's important for a microbiologist to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli when they come from a clinical specimen. In this lab, we're going to talk about several biochemical tests which are common and used to identify gram-negative bacilli. Here we're looking at a very large number, and we're not going to talk about that many. The first test we're going to talk about is the indole test. Some organisms can produce the enzyme tryptophanase that converts the amino acid tryptophan into the products indole and pyruvic acid. They make neutral products. And you can test for that by adding Kovacs reagent, which turns red when indole is in solution. Any question about any of that? All right. So what you do is you overnight incubate the media, which has the amino acid tryptophan in it, or it could be a media with rich protein, so that when the protein is digested, tryptophan is released. And then you have overnight incubation and you add Kovacs reagent to it. Kovacs reagent will turn red at the surface. E. coli tests positive for indole production. Other members of the family may not. This isn't looking very red, but that's actually positive. And I think this is a problem of the photograph. It looks kind of purple, but tryptophan in the enzyme tryptophanase makes indole and pyruvic acid. You would then add Kovacs reagent and it turns red. And that should be more red. That kind of looks purple. My apologies. Like I said, I think that's a problem with the picture. All right, the negative is not red and it's going to be a yellowish color. The next test we're going to talk about is the methyl red test. And the methyl red test is frequently, not always, ran together with the VP test. What you do is you have one media, because the MR media and the VP media is the same, and you take one tube and you inoculate it. And then you grow that tube up, and then you split it into two. One portion is used for the methyl red test. The other portion of the tube is used for the VP test. Both methyl red and the VP test are detecting the end products of glycolysis. The methyl red is testing for acid products. The VP test is testing for neutral products. So glycolysis of glucose makes pyruvate, you know that. It also makes some other end products. If the end products are acid, we can discover that with the MR test, and it'll turn red if acids are present. If the end products are made, we can test that with the VP test. And once again, it turns red if it's positive for the VP test. As a general rule, if the MR test is positive, the VP test is negative, and vice versa, but there are some exceptions. So MR test has to be run for 5 days, because if you incubate it for less than 5 days, and then you do the test, if it's negative, you then have to do the MR test again, start over, and then incubate for 5 days. So generally, as a rule, we in the lab incubate the MR test for 5 days. The VP test only needs to be incubated for 2 days, but it can be incubated for as long as 5 days. Both tests, if they're positive, turn red. With the MR test, you just add methyl red to the test. And if there's acid in the broth from the metabolism of glucose, fermentation of glucose, then the methyl red will remain red. If there is not acid in the tube, the methyl red will change color to yellow. And that would be a negative test. Positive, the tube is red. For the VP test, after you incubate it, you then add Barrett's solution A and Barrett's solution B, shake the tube to add oxygen to the tube, and then let it sit for about 60 minutes. If acetone is produced, which is the neutral product, the VP solution will turn positive. It usually is not all of the tube, but if a portion of the tube is red, then that's VP positive. If the tube remains yellow, then it's VP negative. So negative for both VP and MR is negative. If it's red, it's positive. Any question about any of that? All right, so indole, MR and VP, if it's positive, it is red. If it's negative, it's not red and it's usually yellow or yellowish. Let's move on to the last of the IMVIC tests, the citrate utilization test. Citrate is the only carbon source in this tube. If the bacteria can metabolize citrate, the bacteria will then grow in the tube and produce basic products. The basic products will make the tube change color to blue. So if it's blue in color and you can see bacteria growth, it is positive. If it's negative for the citrate test, generally the bacteria do not grow and the tube does not change color, it remains green. Any question about that? Here we're looking at citrate, and as you recall, that's part of the Krebs cycle. If the bacteria can metabolize citrate, it will turn the tube blue. If it cannot, it will be green, and citrate is the only carbon source in that tube. All right, let's move on to the carbohydrate metabolism test. I think we talked about this earlier. The bacteria can be looked to see if they hydrolyze a specific sugar. The sugar can vary. There's one sugar in each tube. That's why we call it carbohydrate metabolism, because all of the sugars are carbohydrates. You can study glucose, lactose, sucrose, maltose, fructose. I think we do inulin. I don't think we do fructose. And there's a few other sugars you can test as well. If the sugar can be metabolized, it will produce acid products in fermentation, and that will change the media to yellow. If you see yellow color, you go A for, that's how you score this, A for acid. And then you look in the inverted Durham tube there, and you look to see if there's a bubble. If there is a bubble, then gas is produced, and you score this one as AG. Generally, if there is a bubble, there's fermentation going on, and so you'll always see AG or G with A, so you'll always have AG, but you could only have A, where fermentation occurs, producing the acid, but the fermentation does not produce gas, and so you only score that as A. And the other possibility is negative, where the tube is not changed in its color. It is not fermented. The sugars are not fermented. Each tube has one sugar in it, and so you have to ask for the glucose fermentation test or the sucrose fermentation test or some other. Any question about any of that?
Would this result be the same for the PR lactose test that you sent us originally?
Yeah, this will be the same as the PR lactose test. So these are called well, actually, they're usually called, sorry, the PR carbohydrate test. And I don't have that here for some reason, but this is the same. The carbohydrate metabolism test is the PR carbohydrate test. The reason why it's called PR is phenol red is the pH indicator in the media, PR for phenol red, and it's red under basic or neutral pH, and under acidic pHs, it turns yellow. So that actually, I should change that and make that PR carbohydrate metabolism test. I'll have to do that online. But for now, I've got it here. Actually, hopefully I can save this. Since it doesn't have the verification, it wants to shut down the Microsoft. I have until August 20th to get verified. If worse comes to worse, I just take the computer into Clark College, and then it will use the Clark network, and it knows I'm a Clark instructor, and so it's okay. But usually I can get it to work by just logging in, and it figures it out. Meaning I can do it from home. I just have to log in. All right, let's talk about the motility medium. The motility medium is a semi-solid agar that is lower in agar than is normally found. And the bacteria can actually swim through this semi-solid agar. So it's not entirely solid, and that is it's semi-solid. What you do is you stab inoculate with a needle, making a straight stab down into the tube. And if the bacteria only grow on the stab, then it is a non-motile bacteria. If, on the other hand, the bacteria grow everywhere in the tube, and that's why that's cloudy, because the bacteria is growing everywhere in the tube, then the bacteria are very motile. What do you do if the bacteria are slightly motile? Then it doesn't look like that. It looks like this. Here's our tube. There's the semi-solid agar. And then you have the stab mark. The bacteria does grow on the stab, but it flares out of the stab. And the flare usually looks something like this. There'll be different sizes, different pattern on each side. So let's make it like that. Then make a real big one here. And maybe a little one right there. That's the flare. So it does grow on the stab. I'll try and make it dark there, but it flares out of the stab. And that's trying to show you a flare. Any question about that? And if you see that, you know the bacteria is motile. It's just not very motile, because if the bacteria were very motile, it would be everywhere in the tube. If it's not everywhere in the tube, but it's flaring out from the stab, it is motile. It's just slightly motile. And yes, the bacteria have different motility. I probably have a picture of a motile bacteria someplace like that. I should go get it. Anyways, that's the motility media, looking at the bacteria. Remember, if the bacteria are motile, it has at least one flagellum. It may have several flagella, but all prokaryotes can only move by flagella, at least only swim by flagella. You can test for motilla in different media. This is the motility media. You can also test for it in the SIMS media. I'll take a look at that. Let's scroll down here. Keep going. Keep going. There we go. See if I can blow this up just a little. All right. So you can see this has a bacteria in the stab mark. Hopefully you can see that. That also is bacterioma in the stab mark. This one, though, it's flaring out. This is actually a good picture of the flare because it's not everywhere. It's right here. Not much of a flare down here, but a large flare on this side. But the point is, it's not everywhere, it's flaring. That one, the bacteria is everywhere, and that's why it's dark everywhere. So this one is motile. And you can't really see the cloudiness because of the black. And we'll talk more about that when we talk about the SIMS test. Let's see. All right, that's it for the motility test. Let's talk about hydrogen sulfide production. Bacteria can produce hydrogen sulfide. If they produce hydrogen sulfide, then you'll find a black precipitate in all of the tests that test for hydrogen sulfide production. Is that clear? We have different tests that can measure the production of hydrogen sulfide. You can test for hydrogen sulfide production with the triple sugar iron, also called the TSI test. You can test for hydrogen sulfide in the sulfide indole motility test, also called the SIM test. You can test for hydrogen sulfide production in the hectone enteric agar plates, also called the HE agar plate. All of them are testing for the production of hydrogen sulfide, and then they form a black precipitate if hydrogen sulfide is made. Any question about any of that? Some bacteria that form hydrogen sulfide and produce the black precipitate include Citrobacter, Salmonella, and Proteus. Those are genera. So there's a black precipitate, and there is a negative hydrogen sulfide example. Usually, hydrogen sulfide is produced anaerobically, and that's why the very top of this tube is not black, because aerobic respiration is happening here, and so hydrogen sulfide is not being produced there. It's being produced here, and the hydrogen sulfide may have was diffused up here. Okay? But there's the hydrogen sulfide test, and that's probably, what is that? That might be SIMS media. That's not triple sugar iron. So that's probably the SIMS media. You can also test for hydrogen sulfide production in triple sugar iron. You stab inoculate. Hydrogen sulfide, once again, happens in the bottom portion of the tube, turns black. Hydrogen sulfide is produced. Hydrogen sulfide positive and hydrogen sulfide negative, all the other tubes. And this is looking at triple sugar iron. We'll talk more about that in a little bit. This is SIMS test, hydrogen sulfide being produced here. As I mentioned, you can't see it because it's black, but because it's black everywhere, we know that this bacteria is motile, and so 2 is motile. Where is that? Motile positive, tube 2. Control is negative. Excuse me, tube 1 is motile positive. Tube 2 is motile negative. That I already said that and that. I just was reading that wrong. Any question about that? anyways, that's positive for hydrogen sulfide. These tubes are all negative for hydrogen sulfide because they're not black in the Sims tube. And the other time you can measure hydrogen sulfide production is you can grow it on an HE plate. This organism is not black. Those are white or off-white colonies. And these colonies are black. And that's because these colonies are producing hydrogen sulfide on the HE plate. Any question about any of that? All right. Let's talk about the triple sugar iron test. What you do with the triple sugar iron test is you inoculate it twice. You stab inoculate it to get bacteria growing in the butt. That's this region of the tube here. And that should go down about 2/3 of the way here. It looks like it's going closer to 50% or maybe 60%. So 2/3 of the way from here. The reason why you want to go down deeper than this is you want the growth to be anaerobic in the butt. And then you inoculate the slant, and this you do by taking the wire and then putting the bacteria on the slant. And then this area will be growing aerobically. This area will be growing anaerobically. And then you compare your results of the TSI tube. TSI tube has 3 sugars in it: glucose, lactose, and sucrose. But note the glucose is at 10-fold less concentration than lactose and sucrose. It is a pH indicator to determine the pH, and it has sodium thiosulfate in it to detect the hydrogen sulfide production. After you inoculate both the butt and the slant with bacteria, the butt here, the slant there, you then incubate it and look to see the results. Look for the color in the butt and the color in the slant, and you put the butt underneath the slant, and then read it that way. Look for whether there's gas generated and hydrogen sulfide being produced. If there's cracking of the media or a bubble in the media, that means gas was produced. If it's black, so this has a bubble in it as well as cracking of the media, so gas was produced. If it's black, hydrogen sulfide was being produced. Okay, so no gas, gas, hydrogen sulfide, no hydrogen sulfide. You then look at the color in the slant and in the butt. So this is pinkish or reddish in the slant and pinkish or reddish in the butt. And so you put that, you don't need to put it as K, but we'll just say negative, negative for the slant, negative for the butt. So the slant comes first and then the butt. For tube 2, it's hard to read this one, so let me come back to it. Let me go to tube 4 because this is the simplest one. Tube 4, it has yellow in the slant and yellow in the butt. So we say A for acid in the slant over A for acid in the butt. When we have this case, we know that glucose is fermented, and then either lactose or sucrose or both sugars are fermented, both in the slant and in the butt. Gas is produced because there's a bubble, no hydrogen sulfide being produced because it's not black. Any questions about tube 4? Tube 3 is red in the slant and yellow in the butt. So you go negative or K for the slant, A for the butt. This means that only glucose is fermented because the slant is red, the butt is yellow. If there was lactose or sucrose fermented, the slant would be yellow because that sugar is at 10 times the concentration of glucose. because glucose is being fermented. Glucose has been totally used up on the slant because the bacteria grow faster aerobically than they are doing anaerobically in the butt. So all of the glucose got used up. If initially we looked at the fermentation before a day or before 2 days, this is, I guess, the TSI, we would have seen that the slant would have been yellow at some point early on. But now it's red because the bacteria, once they use up all the glucose, that produces yellow because acids are being produced. But then the bacteria, rather than dying, start utilizing the protein in the media. If they utilize the protein, they start making amine and then release it as ammonia off of the amino acids. And then that's basic. That will change the yellow slant to reddish or pinkish. We know that the bacteria was fermenting glucose because in the slant, the bacteria grow slower, ferment slower, and in the slant, it's still yellow. Any question about any of that? So only glucose is fermented when the slant is pinkish or reddish and the butt is yellow. And that's because glucose is at low concentration and it gets used up in the slant. And then the proteins revert the color of the slant to a pinkish color. All right, tube 2, it's hard to see, but wherever a hydrogen sulfide is being produced, that means we have fermentation. So the color of the slant would be yellow. In this case, only glucose is fermented because the slant is pink and the butt is yellow. Although you can't see that it's yellow, we know it's yellow because wherever hydrogen sulfide is produced, fermentation occurred. There's no gas being produced here, and there is hydrogen sulfide being produced here. So this is a complicated test. There's many things going on at one time, and the test is looking at a number of things. There's looking at the color in the slant, the color in the butt. It's looking at the production of gas, and it's looking at the production of hydrogen sulfide. All right, the Sims test is another test where 3 things are being tested. What you do is you stab inoculate the tube, and then you look to see where there's growth. If there's growth only in the stab, that means the bacteria is non-motile. If the bacteria flares out from the stab or moves everywhere in the tube, then the bacteria is motile. You then add Kovacs reagent to the top of the tube. Kovacs reagent floats. And if it turns pink, then indole was produced. And so these tubes, tube 1, tube 3, and 4, are indole positive. The control in tube 5 are indole negative. You then look to see if the tube is black. And if the tube is black, hydrogen sulfide is being produced. So the SEMS tube is looking at sulfur production, hydrogen sulfide. It's looking at indole being produced when you're adding the Kovacs reagent. And then it's looking at motility. So SEM, sulfide, indole, and then motility. That's what SEM stands for. To determine the indole, you need to add Kovacs reagent to the top of the tube, and Kovacs reagent will float over the media. And if it's reddish or pinkish, then it is positive for indole. If it's not yellowish or off-white, it's negative. All right, any question about SEMS? All right, McConkey's agar, also called MAC, and heptone enteric agar, HE, run at least 2 or 3 tests at one time. McConkey's and HE plates are selective for gram negatives. So gram positives tend not to grow on both MAC and HE plates. If you do have growth, you can then assay the growth for fermentation. On MAC, if it's fermentation positive for the sugar lactose, the growth as well as the media will turn pink. If it's not fermenting the sugar lactose, the color will be its normal color, and here it's off-white. For HE plates, if the bacteria can ferment the sugar lactose, it will be salmon color. If it does not ferment the sugar lactose, it will not be salmon color and it'll be normal color. In addition, on HE plates, you can assay for hydrogen sulfide production, and it'll be black if you have hydrogen sulfide being produced. Any question about MAC and HE plates? Remember, you can only assay for lactose fermentation on MAC and HE plates if the bacteria grow. So gram positives should not grow on either plate. So you cannot assay for lactose fermentation. You have to have growth to be able to assay for that. All right, let's go into the lab. You're not physically going to be doing any of these biochemical tests, but you will be held responsible for knowing how to perform and interpret them. We have video clips for you to watch on carbohydrate fermentation, the SIMS test, the MRVP test. Remember, that's 2 different tests, the TSI test, and then the McConkey's agar test. Any question about any of that? Let's go ahead and go to the worksheet now. Nope, not quite. Got to come back here. You also need to view some of the website Microbuzz. Open it and then click on the, well, that's not going to open for me. Open it and then click on the differential test right here. And you only need to do the tests that you need to fill in the table on the worksheet right here. Some of these tests we have talked about in the past. Some of them we talked about in this lab. And I think all of them are on the Microbuzz website. The first one has been filled in for you for the positive result. You do need to put in the negative result for the oxidase test. And we studied that in the last lab. The cell or spot turns blue if it's oxidase positive. So we talked about indole today, methyl red today, VP today, Simmons citrate today. We talked about carbohydrate fermentation. If the sugar is fermented, what's the positive result, the negative result? If gas is produced in carbohydrate fermentation, give me the result. And if it's negative. TSI agar slant, we did talk about it in today's lab. So mention if glucose is fermented, what the positive result looks like. And remember, you have to have the slant over the butt for this. Lactose sucrose fermentation, positive result, negative result. Hydrogen sulfide production, positive result, negative result. This is of a TSI. Gas being produced, tell me what it looks like or what you get for the positive result. And then the negative result, no change in the tube for gas production. Sims agar test, we talked about that today. Sulfide production, tell me what the positive result is, what the negative result is. The indole test, tell me what the positive result looks like, what the negative result looks like. Motility test, tell me what it looks like and negative looks like. And then we have, for some reason, the MAC and HE plates together. tell me what lactose fermentation looks like, at least of the MAC, because it's a different color than HE. Okay, so at least positive for the MAC and then negative for the MAC. If you want both for the MAC and the HE, you can do that. And then complete the following questions. 2A, what reagent must be added to detect the presence of indole? So name the reagent. 2B, methyl red is a pH indicator that remains what color under acidic conditions? C, the what test uses Barrett solution A and Barrett solution B reagents to detect the presence of acetone, a neutral product formed from glucose metabolism. D, Simmons citrate uses the pH indicator. Give me the name of the pH indicator. And it turns blue color under, and you have to either bold or write it out, acidic or basic conditions. I guess you could underline it as well. Okay, you have to distinguish what it is. Either acidic or basic conditions, the pH indicator turns blue. 2E, what pH indicator is used to detect carbohydrate fermentation and is yellow under acidic or basic conditions? And this should be PR, carbohydrate fermentation. Is that right? Yes. So it's yellow under either acidic or basic conditions. You need to bold one of the answers or underline the answer, or you can write it over here. F, organisms that possess this cell structure test positive for motility. So name the cell structure. We're talking about prokaryote organisms, by the way. 3 organisms that produce hydrogen sulfide are, name 3 species that produce hydrogen sulfide. H, if an organism ferments glucose but not lactose or sucrose, the TSI slant will be what color for the slant, and the butt will be what color? And for I, the 3 tests for SIMS is what? Name them. And then MAC and HE plates are both what? And they inhibit the growth of either gram positive or gram negative. You got to answer that. You can bold it, underline it, or write it. And they are also what? In that they separate various bacteria based on visual differences from sugar fermentation. All right, so this is the key you need to solve the answers for the case study in Lab 9. Only use this key for Lab 9. Do not use it for any other lab. Do not use it for your unknown project. Okay, but use it for Lab 9. For some reason, I've had students use a previous key from like Lab 8, and that's going to give you the wrong answers. And then fill in this table for case study 1. Say what the oxidase test was, the glucose fermentation, the lactose fermentation, the triple sugar iron results, the H2S production, that's hydrogen sulfide production, the motility. So these you just say plus or minus. Indole plus or minus, MR plus or minus, VP plus or minus, citrate plus or minus. TSI is a little more complicated, so you might have to put in a longer answer there. So fill this in for each of the case studies, and then tell me which organism from the table, which is either a member of Enterobacteriaceae or Pseudomonaceae, which is responsible for the case studies of case study 1, 2, 3, 4, and 5. So mention a species here. And for the case studies, you got to go back to the lab module. I won't go through them, but I'll just show you where they are. The case study is below. Here we are, case study 1. You can read about it. Here's the different tests. That's the bacteria from the infection of the patient growing on blood agar, growing on McConkey's agar, and that's a result there, by the way. And there's the gram stain. You may need to blow that up. Yep, I'm not able to blow that up here. And some of those look like cockeye, but actually all of them are bacilli in this lab. And then some positive tests, or I should say some tests. Not all of them are positive tests, but some tests that were done. I think this page gets split. So the first one is the SIMS test, the MR test, the VP test, the Simmons citrate test, and then I think that's triple sugar iron. Yep, TSI. So record your results and then mention what species it is. For case study 2, Mikey, a little boy, his mother finally bought him a turtle. He's good about taking care of the turtle, but he's not so good about washing his hands. And then he gets sick, and there's some culture growing on McConkey's plate. And there's the culture growing on HE plate. And those are bacteria of interest. I think the pointer is pointing at a bacteria of interest. And yeah, it denotes the colonies of interest. And then what does that mean? They then do the catalase test. I don't see the catalase test. Where is it? Catalase positive, oxidase negative. And then here's some more tests you have to interpret. There's the SIMS test, a lot of things going on. The MR test, the VP test, the Simmons citrate test, and then the triple sugar iron test. All right, any questions about any of that? If not, you can go ahead and read the other case studies. And if you have any questions, I'll be here until 8:30 to help you with questions on the lab. So go ahead and get started unless there's questions.
So you said that you'd send out the results from the test for the unknowns project later today or tomorrow?
I will send it out later today. I
Okay,
did
great. I
not
just
send
wanted
it
to
out
make
today.
sure
I did not
I
send it out
requested it
today.
appropriately. Okay,
Yeah, I haven't sent out today, but I will send it out later today.
awesome. Thank you so much.
Okay.
I have a question.
Uh-huh.
So this is regarding the lecture. Um, For lecture worksheet, 8B. I got the question, the very last question that was talking about the macro mutation.
Uh-huh.
And, so I couldn't find any information on the chapter notes. So I looked it up online. That's how I got my answer. And I wasn't sure what you were meaning. The question I answered was, what is the macro mutation where an entire chromosome is extra? So I said, I don't know if I'm saying this correctly, but is it polyploidy?
That would be correct. I
Okay.
think what I'm going after is Down syndrome, because that's the one I know off the top of my head. Normally,
Okay.
when a person is born or conceived with an extra chromosome, the person will die. So normally, if you have an extra chromosome, you're not going to survive. And normally, I think that the death happens as a embryo or a fetus.
Mm-hmm.
The person usually isn't even born. But with Down syndrome, and then if you have an extra X chromosome or missing an X chromosome, or the same with the Y chromosome, the person can live. In fact, XYY individuals and XXY individuals
Mm-hmm.
are pretty much biologically normal.
Mm-hmm.
You can't look at them and know they have an extra chromosome or something.
Okay. Should I go back and edit that? Are you looking like, should I put in like Down syndrome?
No. If you got polyploidy, that's the correct answer. You did get the fact that I'm referring to there's an extra portion or the entire chromosome, or that's portion of the chromosome is missing, or the entire chromosome is missing. Like there are some people that are born with only an X chromosome. They
Mhm.
aren't biologically normal. These females are females, but they're not capable of reproducing. I don't know if they're mentally normal or if they're like a Down syndrome patient. I don't know. But there are people born with like only one X chromosome. And then there's people born with 3 X chromosomes.
Mhm.
And I think those individuals are also not reproductive.
Okay. I have another question regarding the unknown project.
Uh-huh.
So I'm not sure about how many, like when we want to request a test at one time, how many can I request?
To follow dichotomous logic, you can only request one test at a time.
Okay.